Thank you, Elisa and Miranda and ALD Alliance for inviting me to speak at this conference. I've been here before. When I come to ALD Alliance meeting or ALD Connect meeting, it looks like I'm back with the family or friends. I've known many of you in my other roles, and I'm delighted to be here. I also want to thank ALD Alliance for what you do for the patients, for the families, bringing together experts, the patients, their families, and industry partners in one room to have an open conversation so you can ask any question and the experts can provide you all the answers is really a job that you're doing so well. And together, as it says in their logo, together we can. So thank you very much for that. I also had the honor of taking care of Aiden. I was Aiden's doctor at Duke, and one of the sweetest boys I've ever seen, and a handsome one at that. And so in his honor, when I heard there's going to be green, so I wore 2 articles in green. But Elisa, just a few minutes ago, around lunchtime, shared with me that Aiden had told one of my partners or co-transplanters that he didn't like a tie. And I had no idea. So I would have been fine without the tie, just with the green shirt. So I'm delighted to be here. Let's talk a little bit about gene therapy. I represent Genetix Biotherapeutics, and we have an FDA-approved gene therapy, which is called Skysona. During clinical trials, it was called Elicel. So, and there is a longer name I don't need to pronounce. So Skysona or Elicel is going to be the same thing. Elicel was used during clinical trials, and Skysona is at the, after FDA approval. You probably got a sense that I've been taking care of patients with adrenoleukodystrophy and other leukodystrophy for many, many years. And it's been a special honor to take care of these patients because the bonds that you make with these families is so special. But there are also heartbreaks. 2 special or 2 real important heartbreaks that I have had in my career of doing transplants for 25 years is the first one, when you have a patient who comes to you for transplantation or for any treatment that has the potential to be transformative, but when they come and the patient are already too advanced, they're too progressed, and you cannot provide that treatment, it's a heartbreak. And the fact is that more patients come or have come who could not be transplanted or could not get definitive treatment. And so in that context, what ALD Alliance is doing in newborn screening is really, really important to get the patients to the treatment before they are too advanced. So whatever options that there are is available to each and every patient. And the second heartbreak is, and this is more in the context of bone marrow transplant, stem cell transplant, is you can get a successful transplant, but it comes with certain toxicities and risk, and patients may not survive. A small percentage may not survive because of complications related to transplant. Now, in the 25 years that so many good transplanters have been working in the field, the outcomes have improved significantly. And we heard earlier today from Dr. Duncan and from Dr. Scaradou of how good transplants have become. But it's also important that we continue to have other options. And so we should look at these options in the larger context. I have some disclosures, as I said earlier, that I am representing Genetix Biotherapeutics, but I'm also a professor of transplant and cell therapy at Duke University, and I have been asked to provide. This is all per FDA guidelines that we have to disclose this. In addition, we also have to disclose the regulatory framework under which I can speak. I can talk about the side effect profile of FDA-approved drugs very easily, but I cannot provide any information that talks about the efficacy that is not included in what is known as label, and I will talk a little bit more about it. So there is a limitation of how much I can talk about the efficacy. FDA doesn't want a representative of a pharmaceutical company to come across as being promotional. So I will provide honest information on the drug that is in the label and all the data that exists in terms of side effects, and that doesn't have to be only in the label, that I can provide the latest information. So we have been committed at Bluebird Bio and now Genetix to be very transparent. We have been doing studies, we have done presentations, so in the last couple of years, you can see we've had a couple of presentations at ALD Alliance and also at ALD Connect. And I'm very happy to report here that we also presented, and that work was led by Dr. Pierpont, from whom you just heard, how to assess patients with adrenal leukodystrophy. So that was in November of 2024 at Child Neurology Society, where we presented, or Dr. Pierpont presented, the neurocognitive outcomes of cerebral adrenal leukodystrophy. In addition, our primary goal is to make sure that patients and their families have all the information they need when they have to make the decisions about whether to do transplant or gene therapy. So we have been very supportive of publication of all the clinical data. And the second on your right, the paper is looking at the risk of hematologic malignancy in patients who have been treated with Skysona or Elicel for adrenal leukodystrophy. We did detailed analysis with Dr. Eichler, Dr. Duncan, and many other investigators around the world, and all of that was published in New England Journal of Medicine about 1.5 years ago. Dr. Eichler and Duncan and others also presented the data on the outcomes of the first trial, which was the ALD 102 trial. Developing gene therapy is complex. It takes time, it requires high science, and it requires perseverance. All of that is shown in this slide, which goes back to about 2011, when early studies were done looking at the natural history of the disease. There were 2 studies, 101 and 103, that were conducted, and then the first clinical study was 102, ALD 102, that spanned from 2013 to almost 2020, and then the second one was ALD 104, which started in 2019 and went on until 2023. Per FDA guidance, these studies were 2 years each, but per FDA guidance, we needed to follow the patients for a longer period of time for safety. So all patients who were treated on 102 and 104 then enrolled into the long-term follow-up study, which is LTF 304. Additionally, once we had Skysona approved, and that's a little star at the bottom you see, around middle of 2022, after that, per FDA post-marketing requirements, we are required to have a registry study for 15 years of some of the patients who were treated with commercial product, and that is the Reg 502 study. There is some data from 103 which looked at the outcomes of transplantation for adrenal leukodystrophy. Actually, my job has been done it's been very easy now this afternoon after this excellent talk by Dr. Duncan Skaraduvu, where they talked about the risk associated with allotransplant, and you can see that there is some risk of graft failure as well as transplant-related mortality with variety of donor options that we heard earlier today. Also, we heard the journey of what gene therapy looks like. So let me take 1 minute to talk about, and I will start from the reverse direction. The treatment itself, which is the last block, is chemotherapy, which is usually, I've been told, is a combination of busulfan and cyclophosphamide in the commercial world, is given to prepare the patient, and then these cells, the schizona, is infused into the patient in the hospital. The patient stays in the hospital for a number of weeks for recovery and then is discharged for follow-up by the transplant team. But prior to that, to make schizona, you have to manufacture utilizing patient's own cells that the lab would insert the missing gene into the hematopoietic stem cells. And then those genes, the inserted gene is able to produce ABCD1 protein, which is missing in adrenoleukodystrophy. So that's the manufacturing and detailed testing that is needed per FDA guidance. Prior to that, we need to collect the cells. And that is what is known as the apheresis process. That requires giving 2 medications, usually G-CSF and Pyrexafor, to the patient to mobilize these cells that they can be collected. And they are collected by a process where you are connected to a machine and the blood goes into the centrifuge and the stem cells are collected. And prior to that are the administrative steps of scheduling these collections. Prior to that, you have to get enrolled. So that's the journey in reverse, and that can take some time. However, we understand that patients with adrenoleukodystrophy, once you make a decision to move forward with treatment, don't have a lot of time, and we will prioritize the use of schizona if there is a patient who requires schizona. This I have already explained on the previous slide, so I will skip, other than to say that this was also exactly the similar process during the clinical trials. And there were 2 clinical trials, as I said, 102 and 104. The chemotherapy regimen that was used for conditioning or to prepare the patient was different in those 2 clinical trials. In the earlier clinical trial, it was busulfan and cyclophosphamide. In the second clinical trial, it was a combination of busulfan and fludarabine. This is the outcomes data that is in the label. So label or USPI is that package insert that you may have seen with any drug. Anytime FDA approves a product, it creates a label with the company. And this information is in the label, which is when you compare the natural history of a patient with adrenoleukodystrophy in the context of MFD-free survival or major functional disability-free survival, you look at the dark solid line, which is MFD survival on those patients who were treated with schizona, which is clearly better than those patients who were not treated with schizona. That led to the approval of schizona. Now the second, or this graph, looks at the Kaplan-Meier estimates of overall survival between those patients who were treated with schizona versus those patients who were treated with allogenic transplant from a matched donor, and then those who were treated with allotransplant from mismatched donor. The 2 overlapping lines that you see on the top are those who were treated with either schizona, which is in solid black, and the other overlapping line there is those patients who were treated with matched donor hematopoietic stem cell transplant. The lower line is for mismatched. So there is a little bit more information here on this slide on those 2 trials. There were 32 patients on 102, 35 patients treated on 104, and you can see their age, median age was very similar, 6 and 7 years. The weight is also very similar. their less score was low, but it ranged between 1 to 9 and 1 to 7.5, but the median was 2. NFS was 0 in majority of the patients, so 96% in ALD 102 and 94 in ALD 103. And then we have long-term data. This is data from 2024, and you can see that the follow-up on 102 is as high as 116 months and a little lower for 104. But overall, at that time in 2024, it added up to about more than 300 patient years of follow-up. So very mature data. This is some data that was presented by Dr. Duncan at the Tandem meeting in 2024. There's only one point I would like to point out here, which is that these two graphs show that every patient who received Elicel in the 2 clinical trials actually had full engraftment in neutrophil as well as platelets. Those are the first 2 measures of success of transplantation. Now let me move to, from efficacy to some of the safety issues. When FDA approved Skysona, at that time, there were cases of hematologic malignancy or blood cancer. We have since had few more cases of hematologic malignancy, and I will provide you information very soon on the total number of cases of hematologic malignancy, which was earlier referred to by Dr. Duncan as being, and Dr. Skaradevu as being 10 out of 67 patients, and that's still absolutely true. So this text is from the black box warning on the label. The key components of it are that hematologic malignancy or blood cancer, including life-threatening cases of MDS and acute myeloid leukemia, have occurred in patients treated with Skysona. It has occurred between 14 months to 10 years after treatment with Skysona. That patients should be closely monitored for blood cancer with blood count every 3 months. And then there are other ways of monitoring for this, including a test called clonal expansion called ISA. This details some of the other information. It's a very busy slide, and these slides would be available from ALD Alliance website in the future, and you can certainly refer to it. The point I would like to make here are 2. One is that hematologic malignancy is a known risk. And number 2 is early diagnosis of hematologic malignancy is really, really important. So by checking the blood count on a regular basis is one of the ways to look for any risk of hematologic malignancy, and that is included in the label. If there is hematologic malignancy detected, then there is a hotline at Genetix that can be called by anybody. And in addition, of course, the patients or patients' physicians can also contact us, and we have been informed of these in almost real-time basis. So here is an update on all the cases of blood cancers that have happened. You can count there are 10 cases. The first row talks about the protocol under which those patients were treated. You know now that there were 2 clinical trials, 102, 104. 2 out of the 10 cases were those patients who were treated on 102 trial. 8 patients were treated in 104 trial. We don't know why that difference is in terms of the incidence of 102 versus 104. We have looked at the data in many different ways. I think there is some association, between the two protocols, the chemotherapy that was used, and other factors, but we can't really make a judgment of exactly what those differences are. You can see the age at the time of treatment with LE-cell, and all these were young children. And then the gap between when they were treated with LE-cell and when they developed blood cancer. And it ranges between 14 months to 10 years. One of the things I can say that we clearly saw is in patients who were treated in 102 trial, the gap between when they received the LE-cell to when they developed blood cancer was much longer. It was in years. So you can see one was, I think, about 9 years and 9 months, and the other one was about 8 years. While in 104, the gap between the LE-cell treatment and development of blood cancer was shorter with 104. We also heard earlier that hematologic malignancy is a spectrum. On one end is myelodysplastic syndrome, on the other end is acute myeloid leukemia being more aggressive. 2 of these 10 patients had acute myeloid leukemia, other 8 had myelodysplastic syndrome. 9 out of those 10 patients have been treated with transplant. 9 out of 10 patients are alive. 2 out of 10 patients had relapse, but they are alive. So we have responsibility to the patients as well as to our partners in the medical centers, as well as to FDA and regulatory agencies. So anytime there is a case of hematologic malignancy, that information is provided within guideline period to FDA and other regulatory agency, and to also the Data Safety Monitoring Committee, which we maintain for our long-term follow-up. All of those were done in real time within the required guidelines, and we have also been updating the physicians, the other partners, as well as the ALD community. We have also investigated this, and we have published those findings in New England Journal of Medicine. So in the next section of the talk, I want to talk about, in response to the risk associated with blood cancer, what have we done in the label or the indication? And we heard earlier today that in September of 2025, we worked with FDA to update the label. We do believe that there is, at any given time for any patient, one has to make a decision on the basis of risk and benefit of any treatment. So following discussion with FDA, there were 2 label updates. One was in 2024 of April, and the most recent one is the August 2025. So I will show you the most recent label update. This is straight taken from the label or USPI. What's in black was in the previous version. What is in red was what was added. So what was added is, let me start with that whole paragraph, and I want to be very clear that I'm explaining it right. So Skysona is indicated to slow the progression of neurological dysfunction in boys 4 to 17 years of age with early active cerebral adrenoleukodystrophy. In the previous version, there was no mention of whether they have a donor or not for an allotransplant. But what was included in this update is without those boys, without an available HLA-matched donor for allogenic stem cell transplant. In addition, There were a couple of other changes that were made, and I will highlight it on the next slide, although the other sections of this are just parts of the label itself with the new black box warning, which I showed you 3 or 4 slides back, and the limitation of use. So the other changes that were made were we updated the gap or the duration between LE-cell and development of blood cancer to 14 months and 10 years as opposed to 14 months and 7.5 years. And we also introduced the concept that there is a possibility of relapse of myelodysplastic syndrome, which is a well-known occurrence in patients with MDS. Even after transplant, some patients will have relapse. With this label update, we shared this letter to ALD Alliance and ALD Connect for further information to the patients and families. So I would like to say here that any treatment has risks associated with it, let alone those treatments that are supposed to be transformative. You're changing the immune and the hematological and other aspect of the patient. Those side effects we need to very closely monitor, continue to come up with ways to mitigate those risks, and continue to go through this process of how best we can make it better. This was the study, which is a registry study, and that's part of our monitoring process. So with that, I think we have a couple of minutes left. I want to express deep gratitude, not only to the patients and their families, but also to all our academic partners and clinical trial teams, many of whom are here, the scientists and the researchers that led to the development of these therapies, and the Genetix family. And I'm very happy to say 2 of them are here, and they might want to raise their hands, Lisa Vinya and Tanvi, who are from our larger organization, and I'm very glad that they are here. Thank you very much. I'm happy to take questions.
You don't need to do that. Real quick question. So where are you? You're a professor at Duke, but it says London.
I used to be in London.
Okay.
That was a long time ago. I did some of my training in England.
Okay.
And so the reason it says London is I'm a member of the Royal College of Physicians of London. So that's one of my, or a fellow of the Royal College of Physicians. And that's, yeah. But I live in North Carolina.
Okay, so you're at Duke.
And our company is based in Boston, Somerville in Massachusetts.
Okay. All right, thank you.
Either I was crystal clear, or I did not get through to anybody.
Thank you so much. Maybe you said this, but was there a difference in the 2 protocols in the dosing?
Yeah. Yeah, so there was some difference between the 2 protocols. First one was the chemotherapy difference, which I highlighted. For the first protocol, it was busulfan Cytoxan. For the second protocol, it was busulfan fludarabine. There was also some difference in the mobilization. So the drugs that were used for mobilization, for the first trial, it was G-CSF alone for about, I think, 1/3 of the patient, and the other, they received G-CSF plus plerixafor. For the second trial, all patients received G-CSF plus plerixafor for mobilization. There was also post-transplant need or use of G-CSF was different between those 2 different trials. As far as the dose of the product is concerned, that was not different. And the manufacturing process was not significantly different.
Is there any work going on to basically target integration so that you don't have that random integration causing?
Yeah. So the question is, if I understood correctly, is about integration of this into the genome, and were there off-target insertions?
Right, so
Yeah,
it's
yeah.
target away from
Yeah,
causing a malignancy.
Yeah, absolutely. So a lot of that has been studied very well. And as I said, it's published in great detail in the New England Journal of Medicine paper from October of 2024. By nature, there is insertion in many different sites. By nature. That's the technique. Those insertions can involve oncogenes, which are genes that predispose to development of cancer. MECOM is one of those genes, PRDM16 is another one, and there are other genes. Important piece here, which I think is really critical, is that not all patients who develop or who have insertion into the oncogene will actually develop hematologic malignancy. Almost every patient who has been treated with Elicel has insertion at various levels in many different genes, including oncogenes, but not all develop hematologic malignancy. So there must be many other factors that prevent them from developing hematologic malignancy.
You had mentioned that there were the two different chemotherapy protocols for 102 and 104. For patients going forward that would be in a position to pursue this therapy, is there ongoing work with regard to the vector that you've been using?
Yes, so I know this, and Dr. Duncan, who has treated a number of patients and others, could speak more to it. But as far as I know, most of the patients who are being treated commercially have received, most or if not all, patients have received the chemotherapy that was used for the first trial, which was a combination of busulfan and cyclophosphamide. Okay, well, thank you very much.